Hey everybody, in this lab activity you are going to do some amazing stuff. We're going to use a program that will allow you to actually see how evolutionary change happens at the genetic level. So to get started, the first thing you want to do is open up a web browser. So open up to Google and type in MEGA. That's mega. Type in mega. So the website you want is this one. MEGA stands for Molecular Evolutionary Genetics Analysis, and it's available at www.megasoftware.net. So if you just click on that website, it's going to bring you straight to the MEGA website. Again, MEGA stands for Molecular Evolutionary Genetics Analysis. And this software program is so amazing. It can be used by the layperson in the comfort of your own home, and it's also used by PhD biologists all over the world. So it's user-friendly and it's so powerful. We're just going to be learning the very, very basics of what this program can do. But this program is used by evolutionary biologists to do some major work on evolutionary relationships among species for all sorts of different genes. It's also used to figure out what kinds of evolution are going on in populations. Now, the latest version that they have right now is called Mega 6. So this is the latest version at the time of this recording. They're constantly putting out new versions. You can see that there are Mac versions. I'm going to be presenting this for PCs because I'm somewhat Mac illiterate. So if you're using a Mac, you might have to do some clicking around to figure out exactly what the differences are between the PC and the Mac versions. We are going to use the older version. I think it's user-friendly, and I'm most familiar with it right now. The Mega 6 just came out recently, so I am going to have you go here. So what I want you to do is click on this button that says Other Versions. So click there, and that will bring you to this website, this page. And I want you to go down here and click where it says Windows 4.1. And again, this is if you're using a PC. If you're using a Mac, you're probably going to have to click up here. But again, I'm not very familiar with the way Macs are working on this program. So I'm going to do this for a PC. You want Windows 4.1. And then it's going to ask you just to fill out this little questionnaire. This is a free program, but you'll need to fill out the questionnaire to download the program to your computer. So I'm an instructor. If you're a student, you can put student. It doesn't really matter. And we're at a university or a college. Now mine says Time Warner Cable. I want to change that. And mine's actually not letting me change it, so I'm going to have to just leave that there. But that's okay. It doesn't really matter what gets put here. They just are trying to maintain a database of who's using their program. Anyway, once you do that, you just want to download Mega. And so then you'll get a pop-up window, and you want to save the file, and you'll just follow the prompts. Now, I've actually already loaded this up to my computer, so I'm going to cancel this out, but you'll just continue to follow the prompts. And I'm going to go ahead and close this window out. And you'll see that my Mega here is already here on my desktop. So when you download the program, you'll get these two icons on there. This is the one that you want. And it says preparing to install, and it will give you these other prompts here. You can just click through those, and I'm just going to go ahead and reinstall it. And if you're using high-speed internet, it should go pretty quick. Now you are going to have to be connected to the internet while you use this program. Okay, once you've installed it, you'll see this little thing here. Just say OK, and eventually this window will pop up. You're going to be going to this window a couple times during this process. So what you're going to do with Mega is it's going to allow us to look up any gene that has been sequenced by any genetics lab in the entire world and that has been loaded up onto the genetics database called NCBI, which is maintained by the National Institutes of Health. Now we're going to start off with a particular gene that we're all going to use as the tutorial gene to learn this program. So we're going to start off with a gene that's been identified as contributing to a person's likelihood of getting breast cancer. And the name of that gene is BRCA2. Now you'll also, after you do the BRCA2 gene, you'll be looking for other genes. So let me real quick before we get started with Mega show you an example of how you can find the name of a particular gene for something. So I'm going to go ahead and open up real quickly just another web. I'm going to go to scholar.google.com. Scholar.google.com is a website in Google that allows you to search peer-reviewed scientific journal articles. So these are articles that are written by scientists and are published in stringent scientific journals, and so all scientists around the world have access to critique those articles. So these guys are where we'll be able to access some reliable genetics articles. So if you just put in something like, let's say I was interested in the gene for diabetes, I might put diabetes gene, and actually they've identified multiple genes that make you more prone to diabetes. So if I put in diabetes gene and I click search, you'll get a whole bunch of peer-reviewed journal articles that will have worked on this. So for example, where it says type 1 diabetes T1D in children. So this is a gene that's been identified as contributing to type 1 diabetes. And so you'll see this where the genes are usually have some kind of acronym, usually some capital letters and maybe a number or two in there. See if you can scroll down, you can find some more. Here's another one, IDD3 or IDD. These are different genes. Let's try another one. Let's see if I put in breast cancer. Breast cancer gene. You know, here it talks about the BRCA2 gene. And here's, you know, BRCA1 mutations. So this is how you could find the names of different genes, because you'll be needing to do your own search on your own genes after the tutorial. Let me type one more in here. Let's type in Alzheimer's. Alzheimer's gene. Okay, so here's the Alzheimer's gene, ALG-3. And there may be multiple genes that they've identified for a particular disorder, you know, APP, so that would be another one. So just to get you familiar with the idea that different genes usually have some kind of acronym and usually with a number. Maybe not with a number. Okay, so let me go ahead and I'm going to lower that right now. So let's go back to our mega window here. So the first thing we're going to do is we have to go to the NCBI website where we're going to find the DNA sequence for the gene that we're looking at. So in this case, we're going to look at the breast cancer gene BRCA2. So what we're going to do is we're going to go up to where it says alignment, click on that, go down to where it says query databanks. So when you query databanks, that takes us directly to the NCBI website maintained by the National Institutes of Health. So you can see NCBI, NIH is National Institutes of Health. Now you're going to want to make sure you go through Mega to get to this website, because if you went directly from your web browser, it would lack this button up here that says Add to Alignment. So you want to go from the Mega website. So what I'm going to do is I'm going to enter here the name of the gene, BRCA2, and I'm going to enter Homo sapiens. And you're going to want to make sure you spell this correctly, because if you have any spelling mistakes, it's going to throw off the search. So what we're going to do is we're going to tell this website, okay, search the DNA sequence BRCA2, that gene, for Homo sapiens. And so any lab in the world that's worked on that gene has figured out the ATCG sequence of that gene and entered it into this website, it'll be able to find that sequence. And we're going to find that sequence for humans. And then what we want to do is ask the question with this activity, how did that BRCA2 gene evolve? What other species was it present in? And which ones is it more closely similar to the human sequence? And so how did they get the sequence of these ATCGs? Well, they used a bunch of biotechnology techniques like PCR, polymerase chain reaction, electrophoresis, and then they stuck the DNA into a machine called a DNA sequencer, where it shines lasers onto the molecule, and based on that is able to actually get the exact ATCG sequence of the gene. Okay, so once you've entered BRCA2 Homo sapiens, make sure this says nucleotide and hit search. So now what it's doing is it's calling up all the breast cancer 2 genes for humans that are in the database. And you can see there's several of them. So if you just scroll down, you can see there's a whole lot. Each one of these entries represents a different genetics lab somewhere in the world that's worked on this gene. I'm just going to pick one of these to work on. You want to pick one that's good size. And you want to make sure it says human or Homo sapiens. Like notice this one says mus musculus. That's a mouse. We don't want that one. We want a human. But like notice this one's only 600 BP. That's 600 base pairs long. That's just a little piece of it. This one's only 1,000. These ones are about 11,000. So that's much closer to having the entire sequence sequenced actually. So this one I kind of like. It says Homo sapiens, so we're dealing with human, breast cancer, BRCA2, and mRNA. You want the gene to say mRNA because that means that this is the gene, part of the gene that actually codes for a protein. So if you remember protein synthesis, messenger RNA is written from a DNA template. So you want this to say mRNA. You want it to be nice and large, so this is over 11,000 base pairs long, and you want it to be the correct species. So this is good. So what we're going to do is we're going to go get the DNA sequence for this guy. And we're going to do that by clicking on this button that says FASTA. So I'm going to left click on FASTA, and now that's going to actually give us the actual DNA sequence. Now it's only giving us one side of the DNA. So remember DNA is double-stranded. We're just looking at one side, so obviously if this one side, if there was a G there would have been a C on the other side. But let's just scroll down and look at this gene. So these are all those ATCG sequences of this gene, and this particular sequence is over 11,000 bases long. Look at that. Wow. Amazing. And this is just one part of a chromosome, and this may not even be the whole gene. This is just what they happen to have sequenced in this entry. Now here's what we want to do. We want to look at the evolution of this gene. How did this gene evolve? So what we're going to do is we're going to just take part of this gene and we're going to do what's called a blast search. So in a blast search, it's going to take this sequence or whatever part of this sequence we choose, and it's going to compare it to every other DNA sequence for anything that's in this database, for any species. So it's going to compare it to all the DNA sequences in the database. And based on that, we are going to be able to download the gene sequence in all sorts of different species and be able to create an evolutionary tree for this gene based on those similarities. And here's how we're going to do it. So I'm going to just copy a small portion of this gene. Remember that this gene is huge. If you select the entire gene, it's going to take a really long time for the analysis to happen because it's going to be having to compare that entire thing, the 11,000 some bases, to every gene in the database. So we're just going to select about a paragraph large of this stuff. So I'm going to hold down my left mouse key and scroll over, and I'm just going to select about that much. It doesn't really matter. You just want to make sure it's not too little. Otherwise, there could be some things related by coincidence. And you don't want it too much because then you'll be sitting here for hours trying to do your analysis. So I'm going to select about that much. Now, you can either go up to Edit, Copy, or a shortcut in PCs is just to hold down the Control key and hit the C. So I'm just going to copy this. And now I'm going to go over and click where it says Run Blast. So I'm going to click Run Blast. In this older version of Mega, there's a kind of a little blooper, a little glitch, where a lot of times the script error thing will come up, and it'll say, do you want to continue running scripts on this page? You can just hit yes, and it may pop up a million times. Don't worry about that thing. It's just a little glitch. It shouldn't affect your results at all. Okay, so now you want to click into this box that says enter accession numbers, GIs, or FASTA sequences. We're entering the FASTA sequence. So now you can either go up to Edit, Paste, or hold down the Control key and hit V, and that pastes that sequence that you copied into this window. Now we're going to scroll down. You want to make sure that Others in our etc. is selected. And down here where it says Optimize for, the default is highly similar sequences. Don't click that one. Click somewhat similar sequences. blast in. And the reason why is you'll be here forever waiting for it to pick only the most similar sequences. So hit somewhat similar sequences, and then you're ready to hit blast. And so when it's blasting, it's comparing the sequence you copied to every other DNA sequence in the entire database for all species. Again, notice the script error happened again. Just hit yes, and you might have to do it a couple times. Okay, and eventually this is what's going to pop up. You may have to wait a while if you're dealing with a gene that there hasn't been a whole lot of work done on, because it may have a harder time finding similar sequences. But a lot of work's been done on this breast cancer gene. So when you scroll down, you should see something with a lot of red. The first entry in here is what's called your query. That's the sequence that you entered there. That's the Homo sapiens. And down here, each of these lines represents a different species that was very similar to the one that we queried. Over here, this is the length of the whole gene that we copied in there. So we copied about 1,600 bases. And if there's a line going all the way across, that means that that particular entry had been sequenced for that entire length that we copied. So notice here, whatever species this was, there's a blank there. So they didn't have anything for this first few bases, but they did for the rest of it. If you see a little green thing here, that means that they got that sequence by using a bacterial clone to do it. But anyway, the important thing is you want to see lots of continuous red. The more red you have, the better your results are going to be. So this one looks good. So I'm going to continue scrolling down. And here's what you're going to see here. So the very first sequence is the one that you queried. Okay. You see some different numbers here. The numbers you want to pay attention to are these three right here. This one, query cover, that means how much of the gene that you entered has been sequenced in other words, how much work's been done on it in other species. So you can see most of these are pretty good. If you're in the 80s, even the 70s, you're pretty good. Your E-value should be 0. And this identity, that means that how much of the sequence of this particular entry matched exactly the one that you queried. So obviously the one you queried is 100% identical to the one you queried. But let's look here. So for example, this one, gorilla. So this is a gorilla, and its sequence is 99% identical to the human one that we queried. That's what that tells you. And you can even, let's scroll down way down here. And if you scroll down, check this one out. I don't even know what that guy is. This right here is a macaque. That's a kind of, it's a kind of primate, like a monkey. And notice that it's 97% genetically identical to the human one that we queried. And even if you go way down here, like Canis lupus is a wolf. And you can see a wolf is 83% genetically identical to the one for the human. Anyway, scroll back up. So what we're going to do now is we're going to start adding these DNA sequences for several species into the MEGA program. And you don't want, if you have tons and tons of species that you're adding, it's going to take a really long time to do your analysis, but you have to have at least 5 to really get a good analysis. So you always want to add the one you queried first. So this is the one that we queried. This one we copied and pasted the sequence into the blast function. Every gene that gets entered by a lab into the database gets its own identifying number. So this is the accession number. So put your cursor over the first accession number, and now we're going to right click, and we want to go to open in new window. So you may need to make sure your pop-up blocker is off. You want to do open a new window because it's going to make your life a lot easier so that you're not having to do the backspace or the back browser or anything. So we're going to open a new window. Now your window that got open might be hiding behind your other windows, which I think mine is. So I'm going to lower this down and look for my other one. Okay, so he's hiding a lot. So actually, I think mine's hiding down here, which you probably can't see, but I'm going to have to go way down to the bottom of my screen and find where he is. So I think he's right here. Yes. Okay, so you may have to do some searching around your screen to find the missing window. It's probably hiding there somewhere. So this is what pops up. So this is the sequence for the Homo sapiens that I queried. And you can see it gives you some information about the lab that entered this information. And don't worry about all this other stuff. This is actually the amino acid sequences coded by that DNA sequence. So in this case, each amino acid has a single letter abbreviation. So the M is for methionine, L is for leucine. Don't worry about that. What you should worry about is just right now adding the sequence to the Mega software. So what you're going to do is very simple. You're just going to come up here where it says Add to Alignment. Click, and notice it says Sequence Data Added to Alignment Explorer Successfully. Hit OK. And now don't get rid of this. Actually, you can get rid of this website. So now close that out. Now notice that this window, which is part of the Mega software, got open. This is called the Alignment Explorer. And if you just click anywhere in the sequence, boom, there is the DNA sequence for that Homo sapien that we just added. Pretty cool, and you can click anywhere in here, but this is the ATCG sequence. Now notice this says sequence 1. We want to go ahead and name this so we don't confuse it with all the other species we're going to be adding. So just put your cursor over where it says sequence 1, right click, and put edit sequence name, and put the actual species name here. I'm going to put, you could call this anything you want. You could say human, but I'm going to say Homo sapiens. And now just click off somewhere. Okay, good. So that one's in there. Now I'm going to lower, not close, but I'm going to lower this window and go back to the original page here where I have the list of all the species. Now I'm going to go down. The next one's another human, so we don't need that. We already have a human. But the one after that is the gorilla. I want to add the gorilla's DNA sequence to my alignment. So I'm going to go over here to its accession number, right click, open a new window, and this one actually popped right up. And again, go ahead and add to alignment. And it says sequence data added to alignment explorer successfully. Hit OK. I'm going to close that out, and boom, check it out. Here's the gorilla's DNA sequence here. I'm going to go ahead, right click over that, edit name, gorilla. Okay, so there's my gorilla. Yay, now I'm going to lower that back to here. Next one, Pantroglodytes. That is a chimpanzee. Now you say, how did I know that was a chimpanzee? Well, because at one point I looked it up. And here's how you can, let's say you get to a species and you have no idea what kind of animal you're talking about. Here's how you can easily find out. You can just remember how that's spelled, Pantroglodytes. I'm just going to open another Google page here. This time I'm going to go to regular Google. And I'm going to click on, I'm going to enter this in here, Pantroglodytes. And notice it tells me common chimpanzee, but even if it didn't do that, I could go to images and I will see what I'm looking at. Obviously, it's a chimpanzee. Okay, so let's add this chimp to our database here. So come over here to the chimp's accession number, right click, open a new window, and add to alignment. Okay, and that's successfully added. Close that out. Boom. Let's rename it. Pantroglodytes. Remember, in a species name, the first word is capitalized. That's the genus part of the name. And the rest of the species name is lowercase. Okay, I'm going to add 2 or 3 more species here so you get the idea. Now I'm going to add this guy, Paniscus, which I believe is a bonobo chimp. open a new window, and add to alignment. Okay, so now I got my panpaniscus. So let's write that in here. Panpaniscus. Okay. And now I'm going to add, this is an orangutan, Pongo abilii. So I'm going to add Pongo abilii. Okay, and adding him. I'm going to edit. Pongo abilii. Okay, so notice these are all primates that I have. Let's add a couple of non-primates so that we can really have a good comparison point here. So I'm going to scroll down and find myself something, because all these guys up here are all primates. They're different kinds of monkeys. I could add them if I wanted, but in fact, this little guy, I believe, is some kind of marmoset. He's like a little tiny monkey. I'm going to go ahead and add him just so that we get a primate that's less closely related to humans than the other ones. Okay, so Calithrix jacus. Calithrix. Calithrix jacus. Hopefully I spelled that right. Where's the backspace button? There we go. And let's see. Let me go find a picture of a Calithrix jacus just so we can see it. Calithrix jacus. There he is. And here is a Calithrix jacus. Very, very cute. Some little kind of monkey here. So that's a Calithrix jacus that we just added. So those are all primates. Let's go find something that's not a primate. Okay, now we're in business. Check it out. This guy, Equus cabellus, that's a horse. This one, Orcinus orca, I believe is a killer whale. Orcinus orca. Let's check that out. Let's see. Orcinus orca. There he is. Yeah, it's a killer whale. So let's add a killer whale to there. Amazing. Are we similar to a killer whale? Do they have a breast cancer gene? Amazing, huh? So let's add our horse first, the Equus cabellus. That's a horse. Okay, adding him. And finally, let's go ahead and add our orca here, our killer whale. Notice he has 84% identity, so he's 84% genetically identical to our human sequence. Okay, killer whale is in there. Oops, and I forgot to rename the other one. So Orcinus orca. So this guy was a Orcinus orca, and the other guy was a Equus cabellus. Okay, so you could keep adding more and more if you wanted, but this is good enough for our demonstration purposes. All right, so now what I'm going to do, I'm going to blow this up a little bit. Now notice nothing is in the right place right now because we haven't done what's called the alignment yet. Although it's pretty interesting, even without the alignment, you can see already how similar the DNA sequences are, even though they haven't been aligned yet. So what we have to do now is we have to modify the sequence. So remember that in protein synthesis, the first codon that starts the translation process is always AUG. That's the codon that codes for methionine. Well, because we're working with DNA, there'll be a slight difference here, but we're basically going to be looking for what would code for methionine in the DNA sequence. And so we have to find that start codon. And so what we're going to have to do is for each of these species, go back and look at the coding sequences. So remember that every gene has a lot of non-coding junk in there, evolutionary junk, introns, that get spliced out before translation. So we got to find at least the beginning of the evolutionary junk and get rid of it so that we're starting with the coding region that actually codes for a protein. So we're going to go to Homo sapiens, we're going to right click, and we're going to go to refer to GenBank. and it's going to call back up that page that we originally had where we added the alignment. But this time, if you scroll down, you want to try to find this right here where it says CDS. Sometimes the lab that did the work didn't enter their CDS, but most of them do. CDS stands for coding sequence. So that's the actual, where the gene itself actually starts, the actual part that gets translated into amino acids. So here this says 228. So that means that the gene itself really starts at base number 228. So let's remember that. I can close this out now. 228. So what I want to do is click anywhere in the Homo sapiens, and I'm going to lower this so you can see this. Down here in the left-hand corner, it says site number. This is where you can enter where that first base of the coding region starts. So we said 228. So I'm going to enter that down here, 228, and make sure you're clicked somewhere in the species you're talking about, and then just press enter, return. That takes me to base number 228, and notice it starts ATG. That's what we're looking for here, ATG. If this was RNA, it would say AUG. So this is your start codon. This is what codes for methionine. So what we want to do is get rid of all the evolutionary junk that happens prior to that ATG sequence. So what I'm going to do is, there's your ATG. I'm going to click on one base before that. And I'm going to get rid of all the bases before that. I could go the slow way and hit the backspace, 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 but there's a shortcut. If you hold down the shift key and find the button on your keypad that says home. So if you hold down the shift key and hit home, that selects all those bases prior to that ATG. Now if you just press the delete button on your keyboard, boom, all those guys are deleted. And now I'm starting with the coding codon ATG. So let's do that for the gorilla too. And its starting base will probably be similar to that of a human's because they're so similar. So we're going to go to gorilla, right click, refer to GenBank. So that calls up the page for this gorilla. Scroll down until you get to CDS. So the base where its start codon is for this gorilla is 234. 230. So the 234th base is where that start codon is. So 234, I'm going to remember that and close this out. I'm going to click somewhere in the gorilla and go down here and click 234, and hit enter. And when you hit enter, it goes to the 234th base, ATG. So I'm going to click one base before that, hold down the shift key and home, and now all that before the ATG is selected, hit delete, and now we're starting with the start codon for the gorilla. I'm going to continue doing that. Here's the pan-troglodytes, refer to GenBank. It's going to pull up the page for this chimpanzee. Scroll down, here's CDS. It also is at 234. Imagine that. So remember that. Click somewhere in the pan-troglodytes, go to 234, enter. Boom, there's my ATG. Isn't it amazing how that happens? So remember, one base before that, hold down the shift key, home, and delete. So now he's on ATG. So I'm just going to keep doing this for all these other species. I'll do one more for you and then I'll pause it while I do the rest. Okay, so right click, refer to GenBank. So this is for this, what I think is a bonobo chimp. Scroll down, CDS starts also at 234. Amazing that all these primates are all starting very similar places, so we have very similar evolutionary junk prior to our start codon. Again, click somewhere in there, 234, enter, do, there's your ATG. Hit one base before that, hold down the shift key, home, delete. All right, I'm going to pause this while I finish these other guys. I'll be right. Okay, so now you can see that I have gotten the coding region, the start codon for all these different species. And notice that the further away evolutionarily a species is, the more different the location of that start codon is. So like for the killer whale, the orca here, its start codon was at base 146, whereas most of the primates were at like 234 or 233. So that just illustrates how much evolutionary junk accumulates over evolutionary time. Okay, we have one more thing to do to process this before we're ready to analyze it. And that is, go down here to this scroll bar and slowly scroll all the way to the end of the sequences. And what you'll see is, notice how when you get towards the end, some species might have a huge gap there or extra stuff, and that could mean either that part of the gene wasn't sequenced in those species by the genetics labs that worked on it, or it could mean that there's extra pieces of gene stuck in there over evolutionary time. So what we want to do is scroll back until there's stuff in here for all the species, and then we want to get rid of everything at the end, because otherwise it's gonna confuse the computer. So what we should do up here in the white boxes, just pick a place around here. It could be anywhere. I'm going to pick right here. I'm going to click that white box. That selects the whole, that position for all these species. And now I'm going to hold down the Shift key on my keyboard and hit End on my keyboard. So usually this is located kind of at the top right of your keyboard. I'm going to hit End. And what that did is selected everything to the right. And I'm going to hit Delete. And so now everything's truncated. So now we got to, everything cuts off at the same place, which is what we want. Now go ahead and scroll all the way back over. Now we haven't even aligned this yet you can see how cool this is. Look at this. Most of these bases are exactly the same across all these very different species. The killer whale has almost an identical sequence to that of a human and a gorilla. The gorilla and the human are almost completely identical. Notice like this position here, again, we haven't aligned this yet, but you can see that this orangutan had a mutation over evolutionary time here, whereas everybody else had an adenine at this spot, this guy had a guanine. Here, look at the horse and the killer whale, both have a guanine, and then some kind of mutation happened in the evolution to the primates where they got a cytosine instead of a guanine, and all the primates inherited that cytosine, etc. So you can see where these little tiny mutations are different. Here the horse, and probably all the hoofed animals, had a cytosine instead of an adenine that all the other mammals had, even the killer whale. So pretty amazing. All right, so we're ready to do the actual analysis on this. And so we're going to use a statistical program called ClustalW. And you can either do it by clicking this little button here, or you can go up to Alignment, and it'll say Align by ClustalW. So what this statistical test does is it does a pairwise comparison. That means it compares every two species on this list together. So it compares their sequences and aligns their sequences based on what it thinks happened evolutionarily. And then it does a multiple alignment where it'll then compare all the species overall together. So it's a statistical test that was programmed into this computer. So I'm going to hit Align by ClustalW. It'll say nothing selected for the alignment. Select all, and we hit OK. There's a bunch of parameters here. If you get really good at Mega, you can learn to manipulate parameters. We're just going to go with the defaults. So we're going to hit OK. And here it's working on this. And this could take a few minutes. This could take a while. This is where it's actually right now doing the pairwise alignments. And that's usually the part that takes a long time. Once the pairwise is done, the multiple goes a lot faster. So you just got to be patient, depending on how many species you have in there, how similar they are evolutionarily, all that kind of stuff. That will affect how long you have to sit here and wait for this to be done. So in the meantime, go do your nails or whatever you need to do. Obviously, you need a high-speed internet, or it can take even longer. So I'm going to pause the video so that we can watch, so that you don't have to sit here and wait for this to be done. So I'm going to pause the video, and I'll come back when my alignment's almost done. Okay, so this is just about done. It's coming right to the end. Yeah, this older version of Mega takes a little patience, but it's all good. Okay, so just about finished here. And even when it gets to the end, it can take another minute just to finish up. But any second here, it should tell us that it's all done with that. Okay, so my alignment is done now. And so if you just click anywhere in this box, I mean, look how beautiful that is. This alignment's done. Everywhere where there's a star, that means that the nucleotide base was the same across all these species, not just the humans and other primates, but even this horse and this killer whale. How cool is that? Whereas here you can see that this killer whale had some cytosines here, whereas the other animals had something else, in this case adenine, in this case thymine. Notice here this little monkey had a mutation that was different from everybody else. So those are point mutations, and that's at the end. So now scroll all the way back and you'll see even more similarity when you get to the beginning of this gene. Sometimes you see interesting things like right here. Notice that there's some space right there, and what that means is that there was what's called an insertion mutation over evolutionary time. So the killer whale and the horse and their relatives lacked 3 bases in this position. Somewhere in the evolution towards primates, they picked up 3 additional bases, were inserted into their DNA sequence. In the case of this little Calithrix monkey, he got an ATC. The other primates, what we call higher primates, our closest relatives, the orangutan, bonobo, chimp, and gorilla, these guys had exactly the same base as us here at G. The T's stayed all the way here and the C's stayed all the way here. So kind of interesting. That's what you call an insertion mutation over evolutionary time, that these animals inherited a whole section of DNA that these guys didn't possess. And sometimes you can go the opposite way. Like you can see something like here. Let's see if I can get it back. Okay, right there. Whereas there was cytosines and adenines in these primates here. Here was a cytosine. There's adenine there, but instead of cytosine here, there was adenine. But for some reason, there was a deletion mutation in this little monkey group here and his relatives. They actually lost 3 bases here from their genome, whereas it was retained in these guys. So kind of interesting. This is pretty cool stuff. So that's only part of it, and for your own records, you may want to take a picture of the sequence. In fact, I'm going to blow up the page just so you can see a larger picture of it, just because it's kind of cool to show people. So if you wanted to take a picture of this, you could take a screenshot, and in a PC, all you do for that is you'd hold down Shift and find the key, or actually on my computer, it's the function key, so the FN, I'd hold that down and find the key where it says print screen, and that would give a capture of this screen here, and I could paste that like into PowerPoint or Word. It's just kind of cool. Here's something else that's cool before we go on. This is the DNA sequence, and you can see how similar that is. So you can see, you know, for example, in this spot, only this little Calithrix monkey had a different base here. He had a thymine, whereas everybody else had an adenine. That's cool, but remember that the genetic code is degenerate, it's redundant. So that means sometimes you can have different codons code for the same amino acid. So if I looked at the amino acid sequences, they'd be even more similar than the DNA sequences. And so you can look at that by clicking here where it says translated protein sequences. You'll see the 1-letter abbreviations for the different amino acids. So if I click here, these are your amino acid sequences. Very, very cool. You know, here's I for isoleucine. N here means no amino acid, so there was actually nothing coded there. But here you can see the amino acids are the same across all the species. And if you just kind of scroll around, you can kind of see where the question marks are. That means that it's missing the amino acid there. Anyway, very, very interesting, because remember, ultimately, that's what we're doing with this DNA, is we're creating amino acids so that we can create proteins. Okay, and so in some species, if there's been enough mutations, they may get a different protein or no protein at all due to these mutations that have accumulated over millions of years, over evolutionary time. But let's go back to our DNA sequence, and we're ready to now draw our phylogenetic tree, our evolutionary tree, to see how this gene evolved in these species. So for this, it's going to be kind of scary, but what you're actually going to do is you're going to come up to the upper right corner and you're going to actually close the screen. That's why it's nice if you already took a screen shot of it so that you can go back and look at it. But I'm going to close this, and it's going to say save the current alignment session to file. Say yes. Now you're going to end up giving this, saving it under a name 3 different times. It doesn't really matter what you call this. I could just put some random keys in here, but I'm just going to call this BRCA2, and I'm going to put my name. It's always a good idea for you to put your name on everything. And you can save it to wherever you know how to find it, desktop. So I'm going to do that. Click Save. Now it's going to say Save Data to Megafile. Yes. Give it another name. This time I'm just going to type in some whatever random keys here. And save. And then it'll ask one more time for you to input the title. So I'm just going to, again, actually in this case, maybe I'll go ahead and type in BRCA23 and hit OK. Protein coding nucleotide sequence data, yes. Open the data in a file in Mega, yes. Okay, so this is kind of cool. So the first thing it does is it gives you this window. Everywhere where there's a dot, that means the DNA base was the same across all these species. Anywhere where there's a letter, that means that guy had something different than everybody else. In this case, he had a guanine, or in this case, there was a thymine, etc. So don't worry, that's not really the screen we're going for. Go ahead, lower these other things. Lower that. We want to get back to this. Now, this looks like our original screen that we had, but notice it's slightly different. Things have changed now. This is how we're going to create our phylogenetic tree. Remember, phylogeny just means your evolutionary heritage. So this is your phylogenetic tree. It's the evolutionary heritage diagram for this BRCA2 gene across these different species. So we're going to go to phylogeny. We're going to pull it down. Lots of options, and if you get really good with this program and you learn about it, you can choose different things. A great book to learn advanced things you can do in Mega is called Phylogenetic Trees Made Easy. You can get it off Amazon. Again, Phylogenetic Trees Made Easy. So if you're into this stuff, you can do whole PhDs just doing this stuff. Okay, so I'm going to go down to this one that says Bootstrap Test of Phylogeny. Bootstrapping is a statistical test. Basically what it's going to do is it's going to create an evolutionary tree and give a statistical number that says how sure it is that of the branching in the tree that it's created. And I'm going to go here to where it says neighbor joining. We're going to stick with the default numbers here. Again, if you get advanced, you can learn how to manipulate these numbers, but we're going to go ahead and hit compute. And now it's drawing our phylogenetic tree. And booyah, there it is. Let's open this thing up. Awesome. This is our phylogenetic tree. This is showing the evolutionary heritage of this breast cancer gene. Who'd have thunk? Who'd have thought that a killer whale, an orca, has the gene for breast cancer? Now, whether it actually causes breast cancer in the killer whale, I don't know. There could have been mutations that happened along the way that gave rise to this gene in humans and other primates. So these numbers here represent your bootstrapping numbers. So this means how sure the program is of this evolutionary relationship. So notice this says 100% here. It's sure that this is the way this goes. 68% here means maybe the human was here and the gorilla here. It's only 68% sure that this is how the order that those guys came in. But this is cool. So what this means, notice there's 2 sets of branches here, here and here. So that means that the horse and the killer whale are much closer related evolutionarily in terms of this gene to each other than they are to any of these primates. And all the primates fall on the same set of branches. So these guys are all very closely related in their DNA sequence for this breast cancer gene. But this little monkey that you guys saw is further related from these other primates. Notice the human and the gorilla are almost identical. There's really no length to the branches there. And the chimpanzee and the bonobo chimp are also very, very similar to each other. But all these guys are very similar to each other. Here's your orangutan, so he's further away related evolutionarily than the gorilla and the chimpanzees here. So for that DNA sequence for the BRCA2 gene, we are almost identical to our cousins, the gorillas and the chimps and the bonobo chimps, and very closely related also to the orangutan, less so to this monkey, and much further away to the horse and the killer whale, but still not that far. This number here, 0.02, represents the number of millions of years of divergence that have taken place. So you can see this is over millions of years that this happened, but not too much distance evolutionarily in the primates. And you know, there's different ways you can present this. If you go to View, Tree Branch Style, you can try different cool things. Here's a straight way of presenting this. You could go and try to do a curved version, which is kind of cool. I personally kind of like the traditional one. So when you're all done, you actually are ready to save this. So you can either do a print screen like we did before, a screenshot, or you can just go to, you can also copy it to your clipboard, or you can save it as an enhanced meta file. I'm going to go ahead and copy to clipboard. And now you can open up a PowerPoint presentation. I have a white background. Here I can just pull this open. Make it a little bit larger. You know, you can even give it a neat background if you want. Now I'm ready to paste this into my lab activity handout. You know, you can adjust the size. If you wanted to save it as a separate picture, you could click on it, right click, and save as picture. And then you'd want to pull this down and save it as a JPEG and give it a name, you know, BRCA2 or whatever. So I'm just going to call it B for now. And let's make sure you save it somewhere where you can find it. And now you can actually copy and paste it into the appropriate little boxes on your lab handout. Now you want to make sure that you're including the name of the gene here so that we keep, because you're going to be dealing with several different genes. And if you really want to get fancy, you could also put in pictures of the different animals that are on here, which is always kind of fun. But anyway, that is how to do a phylogenetic tree using MEGA, Molecular Evolutionary Genetics Analysis. That's your tutorial 1. So for this lab activity, you should paste in your tree for the BRCA2 gene, and then pick 3 more genes and create phylogenetic trees using these procedures for each of those 3 genes. And all you're going to do for your lab handout is paste in the trees, because if you get the tree, I know you did the rest of it correctly. And then there's a couple questions to answer. Hope you enjoyed this, and this gets addictive. It's a little slow going the first one that you do, but then you're going to find this fascinating, because how can you argue that genetic change through time, aka evolution, happens when you can actually look at these DNA sequences across very different species and actually see where the mutations occurred? So little genetic change at the molecular level can equal big change at the phenotypic level.